Thank you very much. This is a great opportunity, and obviously I would very much like to thank Kat, Jen, and Nick, and anyone else involved with this conference for giving me this opportunity to talk about our data. I also see many great collaborators, ex-members of my group, and friends in the audience, and I'd like to thank you in advance for your time and for listening. I am going to talk about cholera, and I'm going to mention my current team. As I said, I can see actually quite a few members of my ex-team in here, but these are the recent team who are sat in the audience, lots of them, who are currently really the engines for the cholera work that we're planning and currently doing. And also, I'd like to mention especially Daryl and Matt, who are really integral to everything that I'm going to talk about today. And I'll mention some other people as we go through the talk. So my talk really does look at both historical and current examples of cholera epidemiology using genomics, and we were, as Kat said, interested in this about 10 years ago. I have a long-term interest in enteric disease, so I'm kind of building on Nick's diarrhea. But this is a whole load more diarrhea than he ever had, and this is definitely not a Birmingham problem. But hopefully we can. But it is an important problem, and so there's a little bit of history and a little bit of detail for those who are not familiar with cholera that I kind of need to fill in before giving you some detail about the work that we've actually been doing. So cholera is, when we first started publishing cholera, I remember when you have that embarrassing point where you have to write, people from the publicity department or whatever institute you're at ask you, what are you going to write, what are you going to tell people? And then also they give you a list of questions that perhaps people are interested in. And one of them was cholera is a disease of the Victorian era. Why are you working on this? And this floored me and had a huge, and this was about 10 years ago, and had a massive impact on me from that point on. And so it's important to have the history as well as the current situation. But my work is really, and that of my team and ex-team is really, and many collaborators, is about the 7th pandemic, and that's the current and ongoing pandemic, and I've got a nice timeline to show you in a second. There were previous 6 pandemics, and they were caused by a biotype of Vibrio cholerae called classical. But most, if not all of the talk, I'm going to talk about biotype L-Tor. And as I say, I'll show you a timeline. But there are 2 bits of information that are really important for those, again, not familiar with Vibrio cholerae, is that the strains that caused epidemic cholera, sort of explosive outbreaks of cholera, are generally almost always associated with serogroup 01. 0139 is an important serogroup, but actually historically kind of came and went. It was thought to be the 8th pandemic, but actually kind of fizzled out. And we have got some work on that, but I'm not going to present that. We're going to talk about serogroup 01 strains. And then the other thing to remember is the toxin. And the toxin is really the key virulence determinant associated with acute profuse watery diarrhea. Although there are other determinants as well. And it's clearly, hopefully I'll tell you, it's not quite as simple as that. So the L-Tor, cholera pandemic 7 L-Tor biotype really defines the current pandemic from the 60s, from Sulawesi when it was first observed, all the way through Latin America in the 90s, Africa and Asia in the 60s and 70s, Latin America in the 90s, and then ongoing with recent outbreaks in Haiti and Yemen, and I'll come back to them later. There was a cholera-free period between the first 6 pandemics and the current pandemic, and we're really interested in this period to define what it actually means to have been cholera-free. But there was one, and that's of note. And cholera isn't a disease of the Victorian era, and it's easy to answer that, especially now in the last few years. 10 years ago, actually, it dropped off the tabloid pages, but certainly since 2010 and in 2016 with Haiti and Yemen, there has been enormous amounts of publicity, quite tragic, about the outbreak of cholera and within, especially the Yemen. But what's important is there are still a large number of cases. It's a disease of the impoverished with good water sanitation, probably good vaccination as well, we can do something. And yet, in actual fact, one of the most important treatments for cholera is this bed here, called a cholera cot or a Bangkok bed. And this bucket here is graduated, and that goes under there. And just a bit like Nick would have seen, the difference here is that you're measuring what goes out, so you can then replace those fluids. And so it's a critical piece of treatment for cholera. This is the water that 16 million people are drinking in the Yemen. And so really, as an outbreak, it's not really surprising. So what really can genomics offer to help any of that? And the answer, of course, as we all face, is it could be nothing. It might be that actually genomics doesn't say anything. But in this case, we think it did, and we think it said something quite powerful. And another bit of history, there's a cholera paradigm theory that we, by necessity early on, we actually grated against. But now actually we're trying to work out how to refine the cholera paradigm theory, because this is a good theory, it's a good model, but it's not universally applicable, and I think that's the key thing. You can't generalize the cholera paradigm theory, and yet the science that went into making it is beautiful, very eloquent, and extremely important, and has been important since the 90s when it was first published. The cholera paradigm says that seawater and environmental factors influenced by weather and account really for the primary outbreak of cholera, the movement of cholera into human populations from an environmental reservoir. And here's a copepod, and in the mucous membranes you can find O1 Vibrio cholera. This is an O1 antibody within the mucus of these copepods. So O1 Vibrio cholera do exist, and if you remember the definition that I showed you earlier about the L-Tor 7th pandemic, then O1 becomes an incredibly important marker. And the idea is that there's a reservoir, environmental reservoir, and a cycling between the primary human host and the environmental reservoir. And then there are secondary transmission events, but this being the primary route of transmission within the theory. So this is an important theory because actually it's influenced everything from basic science to intervention. And so modifying and upgrading that theory with genomic data was really our aim. And hopefully we've tried to do that. And I guess what I'm going to try and do in the talk is to qualify these two statements. One's slightly wrong, the other one is pretty much right. But I'm going to qualify them as they go through the talk. So the key question for us all is why now? Why take a look at cholera now? And there are several reasons. One, because of these questions, which I hopefully will answer, or these statements. But two, because I'm lucky to be a member of the Global Task Force for Cholera Control, because the WHO have initiated a cholera control program, Cholera Control 2030, to try and reduce the overall incidence of cholera worldwide. And that's linked to vaccination and to other ways of really focusing on ways to control cholera. So this was our starting point, and there's some very important photos here. You've seen Daryl and Matt already, but we have Francois Xavier and Marie-Laure from the Institut Pasteur, and so these are key, very close, either members of my group or ex-group, or very, very close long-term collaborators, and I've been very fortunate over my career to have really incredible collaborations, and the Institut Pasteur is so incredibly important because it unlocks with its network, it unlocks Francophone Africa. So really we have Anglophone and Francophone Africa within a small collaboration. And this was really our task. This is WHO figures for cholera between 49 and 15. And what you see here are large circles. Obviously, we see these a lot. The bigger the circle, the more incidence of disease. And in my simple mind, I see three pots here. I see sort of South and Southeast Asia, I see Africa, and then I see Latin America. And you can see that with what would be a reasonable hypothesis with a cholera paradigm theory in its current state would be that strains here, it would be reasonable to say that they'd be more closely related to each other than they would for strains, for example, on the west coast of Latin America. And so we thought what we'd try and do when we look at the incidence figures was trying to understand what genomics told us about those populations. And actually, I'm a molecular microbiologist by training, and so I'm really interested in bacterial physiology, biochemistry, regulation, virulence, and so on. And actually, I thought what we'd find is that all of these would have a very strong geographic, biological signal adapted to different conditions, and we'd have a whale of a time really trying to unpick the circuitry within those isolates that represented different endemic populations of cholera. And one of the reasons that I perhaps was led to believe that was because actually the diversity of cholera by standard typing approaches was incredibly high. And so isolates that caused large-scale outbreaks seemed to be different from each other based on either pulse field, MLE, or whatever. And actually, if you look through the literature for various different outbreaks, their links to other ongoing outbreaks in Africa and Asia, for example, might have been questioned, or it might be concluded they weren't linked. And so there was a lot of confusion based on the typing approaches that they use, either serogrouping, MLE, or PFGE. And it's not that they're not great techniques, they are. It's just that genomics add that much more value to that information. And so that's what we did. We actually started to create networks of people, of collaborators, to try and unpick and explain this genomically. And this is my attempt at a sort of, it's a beautiful tree, but it's the way I explain phylogeny. To some extent, the way I like to think of a simple story, and that is that we thought initially we'd have a tree like this, a beautifully almost symmetrical tree with South Asian, if this was the cholera tree, South Asian, African, and Latin American diversity, all held on defined geographically restricted branches. But we didn't. We found something very different. We actually found an asymmetrical type tree, very little variation within the tree at all, and actually geographically all isolates, the geographic signal was all mixed up across the world. Something very different from what we expected, and this is the actual tree we found. And this was published by a student of mine in 2012. And this is the cholera tree with the root and the tips, and I'll go through and explain it a little bit more, but it looked a little bit more asymmetrical. It's asymmetric. And that's Anka, my PhD student. So what does this tree mean? Well, it looked, in first glance, it looked very viral to me. It looked like a sort of an influenza-type phylogenetic tree. But actually, you have a root here, you have multiple branches coming off this very strong single branch that extends up here. And what's important to say at the moment is that most of these strains were clinical. They're from human clinical cases of disease, and that's really important. And so there's a massive bias towards clinical disease here, and I'll come back to that later. What you can see here, it's a dated tree. You can see some of these little flurries here are outbreaks. Well, this one occurred in the 1990s. These two here in the early 2000s. And if we put the country data back on, they're all purple. And these were actually through a very good collaborator of mine in Kenya called Sam Karaoke. And actually, these were 3 different outbreaks, sequential outbreaks over different time periods of cholera within Kenya. I.e., these strains here didn't go on to cause the cholera in the subsequent years, and cholera is a highly endemic cholera country, and so the idea was that the lack of cholera in one year or the low cholera seasons was just a feature of those strains, not having the opportunity to cause disease, but there were endemic local strains present, and so disease could break out. Actually, that's not the case, and so these strains disappeared, and so did they from all over the world. Only to be seemingly replaced by strains further up this branch here, and then again further up this branch here, which led us to this idea that there were 3 sort of almost like dropping water onto a bucket, 3 concentric waves of epidemiological expansions of a very strong lineage, all of which can be traced back to one single strong branch. And actually at the base of most of these branches are strains from South Asia, which led us to believe very strongly that actually cholera wasn't clinical cases of cholera weren't emanating from local reservoirs around the world, but actually were emanating from a place within South Asia. At the time, we call it the Bay of Bengal, which is probably our best estimate. But actually now I think it's more like the Ganges Basin, and so that's something of great interest. One thing just to point out is that wave 2 and 3 were multi-drug resistant, and I'll come back to that later because there's a bit of a paradox there. because we don't actually treat, a primary treatment for cholera is rehydration rather than treatment, although people who are acutely dehydrated do receive IV antibiotics. But wave 2 and 3 strains are multi-drug resistant, whereas wave 1 strains are not. We found something, and this is probably the most important piece of data that we ever produced, I think, and that is just, it's a mutation rate. We estimated the mutation rate from the root to tip distance of that phylogeny. This was done very early on. It's held up all the way through, and it's worked out, it equates to about 3 SNPs per genome per year. And what does that mean? It means that we can combine that phylogenetic data with really, really tight temporal data, plus our isolation dates, to then spread that tree across the world. And we're going to see a lot of that, I guess, over the next few days, of showing how, in a very dated, very tight sense, these strains have spread. But this was our attempt to do that, and this is a slide that I like to talk through because it gives me a chance to elaborate a bit on some of the things that we've done or are doing. So that tree, that data with the mutation rate and with all the dates of isolation and our strong phylogenetic signal, our first isolates were from Sulawesi here, down here in 61. Actually, I think that was where it was first recognized rather than where it emanated from. I think all data points to that. But actually from Sulawesi, it moved to Hong Kong. And so the second isolates were around Hong Kong before they realized, or before cases were reported around Bangladesh. And many, many cases started to be reported within South Asia. So wave 1 strains then appeared in Africa in the 70s, both in North Africa towards Ethiopia and also down the east coast of Africa. Wave 2 strains followed, and wave 3 strains followed a similar sort of pattern, all traced back to the Ganges, to the Bay of Bengal, and all spreading both east and west. And the tree there that I showed you actually probably in the core genome differed by about 200 SNPs. So this is a clonal expansion of 3 waves of Vibrio cholera globally. But one thing we noticed, and I'll talk a little bit more about this, but this was our one isolate at the time, was a Peruvian isolate from 1991. And as I'll show, there was a big mystery about how in 1991, although there was a huge outbreak, huge pandemics of cholera in Africa and South and Southeast Asia, how this epidemic broke out in Peru in 91, and there's lots of theories. But we had this sort of underlies that. So this was the outbreak in 91, over 1 million cases, lots of deaths, and actually originated in January of 91 in Peru and then spread across and down through into Argentina and all the way up through Panama, right the way through to Mexico. So I've kind of said that. The origin was really unclear, actually, whether these were local lineages. And again, the literature was confused simply by the typing approaches that we used to define it. And even recently, this was, I think, 2016, there have been alternative explanations for bringing cholera onto the shores of Peru, and the reason, in an attempt to explain why the 1991 outbreak was so severe, and alternative explanations for how it got there. But what we had was this one strain from Angola in 1989 that was basal to these blue strains here, which are all Latin American strains. They're the wave 1 strains that cause disease within Latin America. This here was our n=1 theory, and we wanted to try and work out whether this n=1 theory here from Angola, which is at the time we were Angola's Portuguese, it's an ex-Portuguese colony. We wanted to know whether Portuguese speaking and whether there was any direct link and travel between Latin America and Angola. So, but there are other theories about how cholera got there in a ship from China, El Nino events, as I've shown, and local evolution. And as I say, the literature was really split quite dramatically between all of those theories. But one thing we noticed actually was that on this branch here leading to the Angolan and the Latin American strains, there were 2 really important changes to the genome. One was within a pathogenicity island, which is actually characteristic of the pandemic 7 strains. There was a replacement of 2 genes within this island here, very subtle but actually quite important. And then actually this has been used as a diagnostic marker for wave 1 Latin American strains, and we called it WASA1 for West Africa, South America. And it's a large insertion. We don't really know what it does. We think it's phage-like, but that's as far as we've got. And actually, it's a region which is diagnostic of all strains within this branch here. So not only did we have an N=1 study, we actually had two very curious insertions within the genome, and there aren't very many insertions actually within the clinical pandemic choleras. And so what we did was we actually went back to West Africa as a group and actually tried to identify other strains that might actually unravel this story. And so I'll come back to that in a second, but this is the tree that actually we produced by that second flurry of going back and trying to look and recreate and fill gaps with archive samples from both from Latin America, from Southeast Asia, and from across Africa. In total, 45 countries out of Africa, across Africa, and almost all of the South American countries. And what we showed was that in blue here, this is the Latin American, the Peruvian outbreak in the 1991. So our Angolan strain is down here somewhere. So our N=1 was one Angolan strain, and then we had these Peruvian strains. It's very clean in that these Peruvian strains are the ones that caused the mass outbreak within 91. There weren't two strains at the time that caused the outbreak, just one. And we can trace the ancestry back to strains that were circulating within Asia or Africa prior to 91, actually pretty accurately based on the time they were recorded. I'll come back to it, but we also actually discovered quite a lot of other things as well. These green strains here were actually circulating in the 90s within Latin America as well. And actually, the outbreak in Latin America in 91 wasn't caused by a single introduction at all. It was actually caused by two concurrent introductions. And I'll show you, one was from Mexico and moved down through Central America, and the other one was introduced into Peru and moved across and up into Central America. And this is the third outbreak. This is the Haitian outbreak, which is attributed to the UN peacekeepers. Their role by the UN has been acknowledged in moving from Nepal into Latin America. And you can see this in more detail here. These are our strains. This was our African strains. And if we looked at them in more detail, here you have Burkina Faso, you have Côte d'Ivoire, and then these are the strains here causing disease in Argentina, in Bolivia at the time. And what we actually found, those two insertions weren't just present in strains in West Africa. As we traced those strains moving through West Africa, we saw the sequential acquisition of those islands, those mutations, within strains that were circulating at the time. So we had very strong evidence that these strains were circulating in the west coast, in the west of Africa, before making the jump into Peru. And so, as I said, that was this introduction here. I'm afraid you can't see the map. It's bleached out. But this was the introduction here in 91. I mentioned that this strain, these ones here actually were introduction at the same time into Mexico. And the third introduction is up here in Haiti. But if we look at our three introductions into Latin America, these are the samples we sequenced from 1990 to 2015, and you can see the WHO incidence figures here. You can see how our sequencing fits beautifully with, sits under very beautifully the WHO figures for cases of cholera across that time period. Okay, so we have three introductions, and actually that explained, as I'll go on, the majority of cases, of epidemic cases within Latin America. But it didn't explain all of the cases of cholera within Latin America, and I'll come back to that in a minute. But what's interesting is that these were the clinical cases. These are the ones I'm going to call seventh pandemic lineage, the epidemic lineage of Vibrio cholera, the species. And the key is that there's very few SNPs differentiating these strains, but they all of that fit under this little triangle here. So what we did, and actually it's largely through heightened surveillance by institutes like the Malbrand Institute, by PAHO and PulseNet Latin America, heightened surveillance and national surveillance as well. In response to the 1990s cholera outbreak, almost every strain of every case of diarrhea isolates were cultured and kept. And so we were able to actually look at not just the pandemic 7 strains, but actually look at a whole mixture of other strains which fell outside of that lineage. And all of these coloured ones here are strains that actually were from either patients, from environment, or from food, and I'll come back to that. So that's a bit of a surprise. But these ones here are the single pandemic lineage underneath that triangle. Why is that important? It's important because if we look at the incidence data for all of those coloured lineages, and here's the pandemic 7, the triangle, the single lineage here, what we can see is very low level incidence over a protracted period of time. And it was often associated with food, and almost none of those cases led to, in fact, none of those circles led to large local outbreaks. They were all very small, largely very small contained cases of cholera. And actually, if you look at the numbers, actually that's less diarrheal disease cases than Campylobacter and Salmonella over the same time period. You wouldn't even recognize these as a diarrheal disease problem, but you would these because at the same time, across the same population, this lineage was causing thousands of cases a week, whereas the others were causing either small numbers or tens of cases in a year rather than causing mass epidemics. What's interesting is actually all these coloured ones are O1. They're serogroup O1, so they qualify as being, by CDC and WHO definition, as being epidemic cholera. And actually, what's even more interesting is some of these, not many of them, but some of them have the toxin as well, the cholera toxin, so they would be O1 toxigenic, which is enough to trigger alert anywhere currently. So, again, I come back to this. Because we know what would cause this pandemic cholera, that triangle explains almost all of the cholera cases that we could actually sequence, get hold of from archive samples within Latin America, where they're linked to large-scale outbreaks. And we have the context to that because we have the background levels of cholera cases, low-level cholera cases, which fall outside of that lineage. And so we're going to try and move on, and so that's important because this is, as I kind of alluded to already, the case definition by the WHO for cholera, and it's around the O1, O139, by culture or by PCR, and then in certain circumstances, the detection of a toxin. So we actually know from the species phylogeny that all of those things, and it's not that surprising from other, if we think about other bacteria, the LPS operon can be exchanged, the toxins present on a bacteria, on a prophage, that can move as well. It needs TCP as a receptor, but it can actually move around. So that's not so surprising. But what we hope to have done is move from species to serogroup to toxigenic, and then identified a single lineage, that triangle within all of this, that it actually causes epidemics and pandemics. And it's true to say that actually of the ones we've sampled, the seventh pet lineage is the one that actually is responsible for the epidemics. So we've changed. We've moved from a paradigm of environmental reservoir cycles of epidemicity and dormancy, that's the cholera paradigm theory, to one that looks at a lineage which actually has a model which really thinks about human introduction followed by cycles of epidemicity and extinction. And so actually we've got two models. Perhaps what we have is endemic cholera, truly endemic, worldwide endemic cholera associated with low-level cases, and we have epidemic choleras. We actually have two models which are consistent with each other. We also have a global sink here. We actually, all our epidemic strains point back to the Bay of Bengal or the Ganges Delta. Okay, but in terms of the introductions into Latin America, one thing about Latin America that's important to know is that it was cholera-free for 100 years. Prior to the 1991 cholera outbreak, it was free. And so Latin America can be epidemic cholera-free. I actually don't think it can be cholera-free, and I don't think we can control Vibrio cholera as a species, because it is a member of our normal environmental biota. But I think there is actually a chance to control seventh pandemic lineage Vibrio cholera, and that's critical. But what we wanted to do now is take our phylogenetic signal and apply this to another country which most people would call hyperendemic. And whilst you might think that those three introductions are acceptable to explain cholera in Latin America, most of us, I think, who've worked on cholera would probably think that was a challenge to use the introduction of cholera into Africa to really explain all of the cases of cholera of epidemic cholera within Africa. And so we put together this tree, and again with Francois, with Marie-Laure, and Daryl, and Matt, and a whole host of collaborators, some of which are here and all of which will be mentioned later, to look at across Africa. And we have wave 1, 2, and 3 strains, so you can perhaps orientate yourself, because I'm going to recolor it in a minute. But these are the 3 waves of global cholera. These are African cholera strains. And so you can see here wave 1, wave 2, and wave 3 here, but they've been recolored. And they're recolored based on Asia being the leafy green, and Africa here is the soil brown. And so what we were looking at were strains. We used the phylogeographic signal, we used the date of isolation, just again to try and understand how cholera in a hyperendemic country compared to that seen in Latin America. And what we can see, you can see pretty easily, is the transitions here from green to brown, and all right the way throughout the tree. And actually what we see is the ancestral strains tend to be green here, and actually those strains that move into Africa tend to be at the end of this phylogeny here. And it happens again and again, and in fact there were 12, there's here T1 to T12, and we called them transmission events. But these are introductions, 12 introductions into Africa. And here we were less successful in accessing archive samples. That's true. It's very difficult to get samples dating back to the 70s, where you haven't had problems due to electrical breakage or freezer malfunction and so on. And so our coverage was much less, about half the coverage, even the interpolated coverage of Africa, of Latin America. But again, actually in this case, we found almost no exceptions. There are some exceptions that we chased down. There was an outbreak of O37 cholera in Sudan in the 60s and 70s, in 68. And so we chased that down, and that turned out to be associated with a contaminated well that was thought to have religious properties, and a community that had built up around that well. But other than that, we found almost no exceptions. All of the strains that we got hold of, and we didn't know what we were selecting, fitted within this tree. And so you can see these different T events. Here, if we look at our coverage across northern, southern, central, western, and eastern Africa, and then this is a summary of them all, you can see where those transmission events, how they sit between 1970 and here the sort of up to 2014. And what we can see is these early transmissions appeared in the 1970s. These are wave 1 strains, actually lingered until the sort of the late 90s, and so on. And you can go through your T numbers of they've entered Africa and then disappeared, right up to T12, which at the time was the most recent outbreak that we'd seen within Africa. There are gaps, and I've mentioned those gaps. There are gaps where we have outbreaks, we have disease that we weren't able to sample, and some of the outbreaks are interpolated from a very small number of samples designed to represent the outbreak. But what we have done is gone back in modern times and looked at outbreaks. And we know in Calcutta, in Pakistan, and in Bangladesh, when we see an outbreak, very large outbreaks, they're usually clonal expansions. So that gives them some confidence about how much those isolates are representative of the underlying disease at the time. And what we found was something quite interesting. When we looked at the actual introductions of cholera, the 11 introductions of cholera into Africa, what we found is the early introductions, you can see here, the T numbers are named here, T1, T3. The dates across here, if you follow down here, you can see from the early T numbers to the late in wave 1, 2, and 3. And what you hopefully can notice is that there was an introduction, the T1 went into West Africa, down here all the way down to Nigeria. Here T3 was spread largely down the east side of Africa, and same with T5 and so on. What we actually found was that actually not only was cholera, almost all of the cholera that we actually sampled introduced into either the west or the east, and there were 12 introductions. It was roughly every 5 to 6 years those new strains were successfully introduced within Africa. And I think that's really important because clearly there are more introductions than we're seeing. These are the ones that took hold and led to large-scale outbreaks. Having said that, the Institut Pasteur for many years has been doing low-level, basic, passive surveillance of cholera within Africa. So again, we're pretty confident that we've covered most things, but there are gaps. There is actually one example. It's actually of a movement from Angola into Mozambique. And there aren't very many examples in total, but there's one example that I want to tell you, and that was during the Mozambique colonial wars, where it's documented the Portuguese soldiers carried cholera into Mozambique during that time, from West Africa, from Angola, moving into Mozambique. And so there are some examples, but stunningly, there are not very many examples, and this pattern holds very strongly. Does it fit with the epidemiological data? And this is a map from Peru, and it looks at the incidence data for Africa. And you can see here the incidence data is away from what they've defined here as coastal regions, which are very generous coastal regions, to be honest. But you can see it fits with the incidence data. So the reason I edited this question, and I continue to edit this question, is that actually we think we know how it moves in truth, and we actually don't know how it moves. But what the data says is that it's not a passive process, and at least we think that the only explanation for the very rapid spread, both across continents and between continents, is by human carriage of cholera. And we think that actually the diversity, the rapid clonal expansion of a single lineage, all of that points towards human-to-human transmission. But human-to-human transmission could be human fecally contaminated environment to human transmission. I think that's really critical, and that's really a distinction from what we've seen before. And so what we've tried to do is to build on the cholera paradigm theory, and I think there really is an opportunity to distinguish between two different ecologies for cholera. And this is clearly one of them. And actually at CFAS down in Weymouth last summer, they were looking at Vibrio cholera populations exploding within shallow water lagoons during our heat wave last year. And some of them are likely to have been O1 cholera. But so this does exist, we all know that, and it's important. But the key thing is to add this on here. This is the second part to it, which is actually the epidemic cholera and the human-to-human transmission, as well as the contamination of the environment and perhaps feeding back into natural populations. And I think that's really critical, that we actually start to build on this paradigm. Phylogenetically, we think we have a model for this, and that's the thing that we're currently trying to test. Phylogenetically, we think we have environmental isolates that we never see. There's a massive diversity of Vibrio cholera as a species. It's enormous, most of which we never see, and we'll only ever see if we take water samples, that people take water samples and start sequencing, which is something we're doing. But there are isolates, perhaps through shellfish contamination, but that's a guess. Do cause cholera, and actually it's clinically defined as cholera, so it's not necessarily that it's a less acute disease. It just doesn't cause large numbers of cases, which points towards transmission. Some of these lineages that we've seen, and this is a cartoon, by the way, if you hadn't already thought about that, but some of those lineages do cause recurrent cases over multiple periods of time, and we've seen that in Mexico, and we think that is to do with a foodborne contamination of lineages, which again don't cause large-scale epidemics, but do cause acute disease, a severe disease, sorry. And then we have a lineage which is associated with a very different ecology and actually does describe epidemic and pandemic disease. And it's the adaptation of that strain that we're really focusing our efforts in research across the groups that I'm affiliated with. So in all of this, and that's really where I'm going to largely end, but I'm actually going to end on unanswered questions. Actually, most of the data we published in 2017. My PhD student Anka published the first in 2012. And so for us, this has been a 10-year journey, but actually most of the data is relatively recent. I think, or maybe that's just because I'm getting old, I don't know. But anyway, it's one or the other. There are gaps. We don't know why there's an extinction event. There's a geographic transmission and an extinction event. But we're really interested in trying to understand whether that's genetic, whether it's immunological, or whether that's a de-adaptation to an environment. So you just wouldn't find environmental endemic reservoirs of this strain because it's changed, and that's quite important. We recently applied all of these skills to the outbreak in the Yemen. And there were some really good reasons for doing that, because it was thought to be a really nasty, hypervirulent strain of Vibrio cholera. And we were able to show that using the contextual data, and that's what we've got, actually the data from Africa and Latin America is the best set of cholera genomes anywhere on the planet. It simply is. But it's not good enough. It's not good enough. And actually what we need now is to really think about where to sample and fill some of those gaps in. And so we've started that. And in the Yemen, it wasn't a new emerging strain. And I work on a lot of bacteria, and I've heard this many times before. The explanation for enormous outbreaks is always the bacteria is a change, and it must be that. In this case, it's not. And actually, the strains, yes, they're wave 3 strains. They're recently emerged strains. That's true. But they were seen first in 2012 in Calcutta. We also then traced them through East Africa before moving up into the Yemen. And the only reason we couldn't be forensic about that is because many countries in that region don't report cholera, but do report acute watery diarrhea, which means that we couldn't sample. But actually, we know with high confidence and through fantastic collaborators like Sam here, we were able to identify the cause of the strain. And actually, it turned out to be almost pan-susceptible to antibiotics, unlike wave 2, all other wave 2 and 3 strains. Actually more treatable, not less. And we've also done something recently. We're going back to Latin America, and there are two very special people in the audience who are pictured here. There's Josefina and Isabel, and Matt's not here, he's in Boston at the moment. But we're going back because actually there's a curious bump here. We looked in Latin America in the early 90s, and we could have left it there. But I'm really intrigued to know what happened in Latin America, and I think it's a one-off opportunity to really understand something very complex about a really important diarrheal disease that will have generic implications for other diarrheal diseases. So we're looking at these little bumps here, and actually it turns out these are strains, these are legacy strains from the original outbreak. They're not new endemic strains or new introductions, they are legacy strains from the initial introduction. And so we have an opportunity to really understand the evolutionary biology of cholera over quite a protracted period of time from a point source introduction into an entire region. And that's with Josefina and Isabel. And we've just come back actually from, I've just come back from Argentina, they live there, where we met the Secretariat of Health and actually used the genomic data to, this is through Josefina, Josefina used that data to convince the Secretariat of Health to change their national health policy for testing diarrheal disease samples. Real power in that data. We also don't know why cholera is drug resistant. Actually, to erythromycin, to all sorts of drugs, some of them not recommended, some recommended. Sam Karaoke has been finding ESBL and carbapenem resistant cholera strains. What we don't see is that those newer antibiotics, so far we haven't seen a lineage that carries them that spread broadly. But we do see local emergence of drug resistant strains. And actually the pandemic 7 strain is multi-drug resistant, but that's been fixed since wave 2. It's penta-resistant. And what we can see here in one of our studies, we looked actually in early, these are the blue colors here are the early introductions into Africa. And we can see that wave 1 strains were, the blue colors were down at 0. They didn't have this plasmid, which is a multi-drug resistant plasmid. They didn't have the SXT, which is an integron that carries similar resistance genes to the NKC plasmid. They didn't have mutations of PAR-C or gyrase associated with drug resistance when they first arrived. But after about 5 to 10 years, you start to see the acquisition in Africa of the plasmids and those drug-resistant markers. However, when these were replaced by wave 2 and 3 strains, those early wave 1 strains, they disappeared, and almost all, in fact, all of the strains that are now introduced into Africa already have SXT. They already have the gyrase mutations. They already have PAR-C. They're already multi-drug resistant before they even enter Africa, apart from some very local quirks that we're focusing on at the moment. We're also going back through collaborations at the ICDRB, and I'm going there just after this meeting. We're focusing on the Ganges Basin and trying to understand really what the Ganges Basin genomically offers to understanding this story, and we really don't know. And it's something that through collaborations both in Bangladesh, but also in the northern regions of India through the Postgraduate Research Institute and Neelam Taneja and my postdoc Alice Taylor-Brown. We're going to try and understand how these strains have emerged and spread across through Bangladesh and India. And these are the collaborators that we're doing that work with. I'd also like to say we're trying to access, do something else. We're actually trying to access modern models or use novel models, and then we have Chris, Leanne, and Paul within my group that have been developing and reapplying actually models, human organoid models. So these are intestinal organoids, beautiful bundles of cell with a hollow lumen that have beautiful structure, really fantastic structure, cross-sectional, little microvilli, Paneth cells. You know, they look gorgeous. And what they've been doing is injecting Vibrio cholera into the Middle East and cholera toxin and watching these organoids swell as you get fluid influx into the actual lumen of the intestinal organoid. It doesn't do everything. Actually, it doesn't replace the mouse or the rabbit model, but it does allow you to get closer to a direct and very fine-tuned human response to different lineages of cholera. And then a little bit indulgent, but I'm here with my good friend Zam Iqbal. We have actually two opportunities where I showed my team because they're young, very motivated people who really are the lifeblood of any group. Just like most of us sat here, we recognize that as a statement that describes all of our groups. And we want people to get involved with the type of work that we're doing, and we have in my group, I have existing SPOD fellows, but we'd really like to extend that, and we have a vacancy really for turning back time, trying to recreate ancestral structures within horizontally transferred elements using synthetic genomics, and so Zam's here with me, and so that's a little bit indulgent, and the rest is that I'd just like to thank the people that really were involved in all of that. Sample collection, data collection, amazing collaborative conversations, and point out, as I say, these are the pictures I've mentioned already. Josefina's in the audience, but we have Francois Xavier and Marie-Laure. We have Daryl and Matt, the two Ds, and then we have just a whole huge number of people. Natalie from Paraguay, Isabel's in the audience, John Clemens, so the people from ICDRB, and many other places across the world who've contributed to these studies. Thank you very much.
As you said, the Vibrio genus is quite diverse, and we know there's also a huge diversity of O antigens, but the seventh pandemic seems to be quite predominated by the O1 antigen. So I was actually wondering, is there anything special about the O1? Do we know if do we know serotypes for previous pandemics?
Yes, we do. So that's a great question, and actually partly I missed that information out because I excluded classical. So O1 is the key marker for the previous 6 pandemic. So it actually turns out that the previous 6 pandemics were caused by a classical biotype of O1 toxin positive. It turns out that that's a single lineage too. So in a way, history has repeated itself. The previous 6 pandemics, as far as we know, and these are people looking into formaldehyde-preserved intestines and stuff, we know that the previous 6 were caused by the classical biotype. It's a single lineage just like this, and that's O1 as well. So it's clear that O1, you wouldn't argue that O1 wasn't important, and there's some nice infection studies that show some importance of the importance of O1. What's interesting is actually our non-classical lineage, our non-7th pet lineage. There are many strains, some of them associated with disease, which are also O1, so you've kind of got to feel that the serogroup is important for disease, but we don't fully understand that yet. We have an opportunity, I think, now, a better opportunity.
Thank you for a good talk. I was thinking back to a paper in the early 2000s from Andy Camilli's group, where they looked at cholera going in and out of people. And apparently when it came out, the gene expression profile, microarray determined in those days, was completely different. And I'm wondering now whether the pandemic lineages that you're sorting out from the O1s, maybe they can do this expression profile, whereas the others can't. Are you looking at, what's turned on in humans?
So again, that's a great question, and I guess it fits with a lot of other enteric pathogens where you have this hyperinfective state of fresh isolates coming out in fresh stool. So I think it's not unreasonable to think that that's the case with cholera, and that might explain why you get this sort of, once you actually establish a number of infections, why you get these explosive outbreaks once you've reached critical mass. So we're part of the work that Alice is doing is to really focus on stool, both of infected and apparently healthy. We're trying to identify carriers, but that's really very difficult. And so we're really turning our attention towards that. We're working with a lady called Jenny Ritchie from Matt Waldor's lab, who re-established the infant mouse, or established the infant mouse model, and is currently working in Surrey. And so we're really working with her to try and answer some of those questions, and we are seeing some interesting stuff.
I have a genomics question. So how much traction are you getting in the wider cholera field for including that lineage in the definition of pandemic cholera?
Well, it's
and is it easy to identify in the field?
So the traction. So as I say, a week ago I was within Buenos Aires, and I was lucky to be invited by Josefina, who sat there, and she's the head of genomics at the Malbrand Institute in Buenos Aires. And very closely linked to the Secretariat of Health, and they changed their policy based on the definition and based on sampling. So I think that there's a danger, and actually I'm part of the lab working group of the GTFCC, and one thing that's really interesting and I'm very conscious of is that without cholera is a neglected disease like many others. We're still talking about a vaccine, and it's the vaccine that Jan Holmgren really pioneered, decades ago. And we're still talking about a lack of molecular tests for cholera. We're still all sorts of things that we don't have. This would resonate with you and typhi and many other diarrheal diseases. They've been neglected. And so the danger is that when we change a definition, you're in essence, you're asking people to stop doing stuff. And that's not what we're doing, because actually in the absence of really good tools, and I think which I think are here now, they just need to be distributed, then people who are doing the serological tests, if they can get the dipsticks, which are not very good unless you use peptone water to amplify the strains, or using culture, which of course is costly and requires some infrastructure, then you need to carry on doing that, because that's the only way until we can replace it. That's the only recommendation I would have.
So maybe a rapid diagnostic.
A rapid diagnostic would be perfect. Absolutely, absolutely.
Hopefully,
Cheap.
more use.
Yeah.